I got involved with AHC because I had a mouse mutant that I was working on that had a mutation in the same gene that's affected in AHC. Although I had this mouse several years ago before it was known that that gene was actually mutated in AHC. So I had this mouse for quite a long time that I knew had motor dysfunction and seizures and cognitive impairments. And then when I saw a paper that came out a couple of years ago that said AHC is caused by mutations in ATP1A3, I thought, at last, I know what my mouse has got. So now what we're doing is actually trying to use this mouse in order to understand more about the what's going wrong in AHC. We first got the mouse when I was actually working in Toronto at Mount Sinai Hospital in Toronto. And actually we found the mouse. We were doing what's called a mutagenesis screen. I won't go into details, but what we were doing is screening lots of mice whose fathers had been given a drug that induces mutations, and basically screening all these mice to find ones that had kind of neurological abnormalities. And so we had one mouse amongst several others that had a kind of neurological abnormality, and then when we crossed that mouse to wild type mice, half of the pups also had this same movement disorder and these other problems. So we ended up calling that mutant line Myshkin, so it's the Myshkin mouse, and then eventually we found that it had a mutation in ATP1A3. And we first really studied that mouse in detail because of its seizures. I mean, at the time, AHC, as I said, it wasn't known that it was caused by mutations in ATP1A3, but epilepsy is a big problem and it's a common disease. So we actually studied the seizures in the mice and published papers on that. But since the knowledge that AHC was caused by mutations in ATP1A3, now what we're doing is actually using that mouse as a model for the disorder. So, for example, what we can do, we can actually take brains from the mice and kind of study what proteins might be abnormal in the brains, or how the cellular structure of the brains might be different. And obviously that's not something you can do with patients. And the other thing we can do is test potential new treatments on the mice. So the mice have symptoms that are similar to the humans. If a new treatment works in the mice, then that's an indication that it might be suitable for testing on patients. And we know from the patient groups that there's like a huge demand for some sort of treatment or cure for this condition because it's so severe. I initially started working on this at, as I said, at Mount Sinai Hospital in Toronto. So I was a postdoctoral fellow there, although I am English originally. So when it came to setting up my own lab, I wanted to go back to England, and so I got a job at Leeds University in Yorkshire, and so I've been there for the past 5 years, and I took those mice with me from Toronto and established a colony of them at Leeds. It's basically one what we call line of mice. So it's not really a strain as such, but it's like a line of mice that carry this mutation. So we've got these mice in the animal facility there, but they are quite fragile though, and it can be quite difficult to breed them, and they have to be treated sort of very carefully. So basically, as a sort of safety net, we've actually frozen down embryos, and they're kind of kept in a sort of special store, just so that we, don't lose the line, or if we did, we could always go back to these frozen embryos. And I suppose the other thing is that we can send these frozen embryos to other labs in the world. So I've already sent those embryos to the lab at Duke University, and we're going to soon send some embryos to a lab at Northwestern University in Chicago, because there's people in other labs who are also interested in AHC, who might be able to do different kind of tests than I can do in the mice. And and the main thing is, as was said at the meeting, is that we should all kind of work together and share our resources to try and ultimately find a cure for this devastating disease. The next steps are always dependent on funding. So we're always trying to get money for our work. And the thing about mouse work is that it's quite expensive to do, particularly behavioural tests on the animals. You have to breed quite a large number of them and keep them in cages, and that can be over several weeks, and that can be quite expensive. So fundraising is important, but also then if we can get the money to do this work, what we'd like to do is actually use the mice to test some new treatments, and that's going to take two approaches. So one approach is, we know that the mice and the patients as well have got a faulty gene, and that faulty gene produces a faulty protein. So one therapeutic approach is to do what's called gene therapy, or at least try and do gene therapy, where basically we put in an additional copy of the normal gene, and we hope that will kind of compensate for the presence of this faulty gene. In the Myshkin mice that I've got, we've already got some evidence that might be successful. But what we'd like to do is develop it in a way that could potentially be translatable to patients. So that's one approach. The other approach is to try different drugs on the mice. And those drugs will be, the selection of those drugs will be sort of driven by studies that other labs are doing. So there are other labs that are working to see which drugs actually affect the activity of this protein, of the sodium pump. And so, they're going to screen hundreds of different compounds, but maybe, the ones that they think are their best 10 hits, then we could then try in the mouse. And I'm pretty optimistic that, over the next 5 years, we'll have a pretty good idea of whether we can develop the transgenic approach or the drug approach, and maybe both will work. So I'm quite confident that there's light at the end of the tunnel. As I said before, the Myshkin mouse, when we first discovered it, it had a mutation in ATP1A3, and then when the human genetic findings came out a couple of years ago, obviously I looked through the list to see, are there any AHC patients that have the same mutation? And in the original paper, there was one patient that had a similar mutation at the same site in the gene, but it was a slightly different change. But actually just today I learned that actually 2 patients have been discovered, one in China and one in Belgium, with AHC that have exactly the same change as in my mice, in the Myshkin mice. So that actually, I think, makes them a better model of the disorder, because in the past those animals had been criticised for the fact that although they recapitulate some of the symptoms, because they apparently didn't have the same mutation as been found in AHC patients, then they weren't, they weren't that good a model. So I feel kind of more optimistic that it's a better model now, and because of that, then that would hopefully, persuade funding agencies to support my work on that mouse.
If I were, on behalf of the AHC Association of Iceland, if I were to say, okay, we have 10,000 euros, is that something that you could work with? Is that something that you could actually make a small project about?
I think it's something that would help. So because in science we either have to take on students, or we have to, who usually don't get paid, they get a stipend to do work, or what we do is actually employ people. And so when you employ people, that's when it gets expensive.
Exactly.
Because you not only have to pay them like a living wage, but in the UK you have to pay like national insurance and kind of other sort of benefits as well. That's when it gets expensive. And so in that case, like, $10,000 or whatever wouldn't really be enough.
Okay.
But for projects that could be actually carried out by a student that would be supervised by me, then in that case that sort of thing is feasible.
Okay.
Because basically essentially the student works for free,
yeah.
But what they get back is good training, and they'll get a degree as well at the end of it.
Exactly.
And then obviously for what we get is a good free pair of hands, but ideally someone that's kind of bright and enthusiastic. And so those small pots of money, what they do is that they help us generate the pilot data that we need to get the big money.
Exactly.
Because the thing about and I've had this in the past with trying to get the bigger money, is that they want you to have either like really good publications, or if you don't have that, then really good solid pilot data. Yes. That you can put in the grant, then that pilot data might even be publishable in itself. And so the smaller amounts of money, like as you said,
money,
that's really where they come in useful, to kind of give us the money to basically give us the resources to generate that pilot data that we need, and then we can go to the bigger agencies. So in the UK, that would be like the Medical Research Council or the Wellcome Trust, and say, we've been supported by a charity to generate this pilot data. Now, will you give us a bigger support to actually take this work forward?
Yes, and that in that case, you could hire professionals to
yeah,
do the
that's
work.
right. I mean, in that case, we could then afford to hire somebody that's got a PhD who would then basically run the project.
Exactly.
And they'd be hired for like three years and could do a lot of work in that.
Yes.
In that time.
Exactly. So when you say large pile of money, what are you talking about? How much would it take for you to actually take a huge step forward?
So at the moment, we don't know which step is going to be successful. So what we need to take, and probably each lab working in the field can take a different approach. So I think what I would like to do is to try this viral vector mediated transgenic rescue, at least to try that. So for that, I think I would need a postdoc to do that, and I think for that the postdoc would have to be employed for at least two years.
Yes.
So, I mean, in that case, in terms of pounds, you'd be looking at £50,000. That's their salary and kind of add-ons per year.
Okay.
The animal costs can be quite expensive. So that could be like £1,000 a month to house like lots of mice.
Yes.
So you can see how it kind of builds up.
Yes.
And then usually the way it works in the UK is that the university want to get some payment for the provision of all the buildings and everything and the support staff that go into supporting these projects. So then they put on their bit, and sometimes that actually doubles the grant, you know. And so then you're looking at hundreds, I'd say at least £200,000, which is a lot of money.
Yes.
And especially when, with diseases like this, that, it's not that long ago that we found out what the causative mutation was. There might be some avenues that kind of just don't work out,
yes,
unfortunately.
And there will be.
I'm sure. And so I think I know that for small charities, if I was going to ask for money, I think I'd feel like I needed to ask for money for things that I thought were probably most likely to kind of work out, and then the more kind of risky things I could put on the bigger grants.
Yes, exactly. Yeah. So, but I was thinking that now, since you today discovered that your Myshkin mice actually have an AHC mutation that has been found in 2 other children, then you would probably have a better likelihood of getting a grant.
I hope so. Yeah, I hope so. I mean, certainly in the past, that's one thing. So when we apply for grants, particularly to the research councils, they go out for what's called peer review. So they get sent to other scientists working in the field. We don't know who they are. They're anonymous, and they make kind of anonymous comments and then give your grant like maybe a score out of 10 or something. And so because, if an animal model doesn't recapitulate the, all of the symptoms, or doesn't have the same kind of mutation, then it's a kind of easy thing to criticize. So for me, I already knew that the mice had a phenotype that recapitulated a lot of the symptoms. And we know that AHC is quite variable anyway.
Yes.
And so it makes me a bit more confident that that criticism won't be one that people will be able to make anymore, because I can say, well, actually, it has been found in 2 different cases in 2 very different countries.
Yes.
I think the main thing for me is that I actually find it encouraging and heartening that that patients groups actually come to these conferences, because for me that's been quite rare on other diseases that I've worked on.
Okay.
And so it actually and to hear you talk about your children and the pride you've got in your children, but also the difficulties that there are, it sort of it really motivates us to try and do the best that we can to try and help you.
Well, as you said, it's a really devastating condition that we live with every day, and so we are ready to do whatever it takes to help our children. Yeah. Yeah, absolutely. I could. I think an obvious thing would be to possibly make it into a PhD studentship. I think that would be a kind of obvious thing.
So to basically fund a PhD student, I think that would be kind of financially kind of feasible in that case.
Because we've been watching you since Rome, and we have a strong feeling that you are someone who could actually be of a really big help for our society.
I hope so.
So that's why we're asking if a seeding fund would be something that you could work with and could give you a little bit of a step forward, so that you could maybe go for bigger things.
Yeah, I think either a PhD student, or if it was a smaller pot of money, like a master's student, which would be like a one year,
yes,
where you wouldn't, they'd have to be paid a small amount, but but then there would be some funds to do the experiments. I think that would be a help to just to really get that extra data that is required. And partly I think part of the difficulty in actually persuading these people to give money is because it is a rare disease.
Yes.
And I think when these panels like score grants, they kind of, these days they're encouraged to say what impact this research will have like on society.
Yeah.
And so if it's a rare disease, then perhaps on society as a whole it might not be such a big effect, but I think what we need to emphasize is how devastating this disease is to the children and their families, even though there might not be that many of them.
Exactly.
But I think the effect of it being a rare disease is that I think the funding agencies need that little bit extra sometimes, maybe even more kind of pilot data to convince them that this should be supported than maybe more common diseases, which I also work on, like schizophrenia,
yes,
where I think they'd be more likely to say, well, let's take a punt, let's just, give him the money, and if nothing comes of it, well, we tried.
Yeah.
But I think with a rare disease, because the pool of potential beneficiaries is quite small, even though the benefits would be huge, then I think sometimes they just need that bit of extra, of nudging in the right direction.
Yes. That's one of the reasons why we're doing the documentary, because if it's a 1 in a million condition, and some, like a publication that came from Denmark, they are suggesting that it's a 1 in 100,000, then we could have 8,000 or possibly 80,000 patients in the world which are undiagnosed. And so with doing the documentary and having subtitles in a lot of languages, then you are reaching to the people, to the doctors who are possibly misdiagnosing their patients.
Yeah, and we've seen from actually the conference this weekend, although unfortunately the lady couldn't attend, that there's work going on in China now, and a huge Chinese population.
Exactly.
I think people would predict that it's actually not really 1 in a million. It's probably more common than that, and
might be.
And it's actually good that countries like China are actually starting to work in the field now.
Yeah. Be very interesting to see your mice and see your what you're doing, and in the meanwhile you could perhaps think of a project that could fit a seeding fund like we are able to provide. Yeah,
and I'll look into the kind of administrative side of what would be the most suitable sort of person to do that. At the moment, I'm thinking probably like a master's student, and then I'll ask the university, how much what would their stipend be and stuff like that. But I think that would be the most sensible thing at the moment.
Okay.
So yeah, From the so at the moment, actually, I don't have anybody working on the Myshkin mice.
No.
But from the 1st of October, I'll have somebody working on them. It's actually in combination with a company who makes sort of a dietary supplement that could be an anti-epileptic treatment, because the mice do have quite severe seizures. Yeah. And so it may be that treatment may also improve some of the other symptoms as well. I mean, it's not the reason for actually looking at the treatment, but
it might be a side effect.
Yeah, that's right. It might be a good side effect.
Yes.